start talking about the lecture then. So, I'm Dr. Casson. I'm a plant molecular biologist, developmental biologist. I'm interested in how the environment regulates plant development and the impact that then has on plant water use, nutrient use and their growth and development. And I'm going to be taking quite a few of the lectures in here. But today's lecture is really more about what you'd say traditional methods in plant breeding. So much of what you'll hear about across the course will actually relate to GM technologies, but you have to appreciate that actually plant breeding has done an awful lot for the world already without ever requiring GM technology. So what we're going to cover is some of those aspects, but of course you can apply these same methods to GM as well. So if you've downloaded the notes, you'll always see some learning outcomes associated with it. I've put some questions on my page for you to look over. These are for your own self-assessment, okay? So it's to see whether you're understanding the material. As I say in the question piece, how much detail you want to go into is entirely up to you, okay? So today we're going to be talking about plant breeding. Because really, when you talk about plant biotechnology, this actually oversees the whole process. So we're first going to look at some plant breeding pipelines before having a quick look at the Green Revolution, which I know Professor Gray mentioned in the first lecture yesterday, and then some other aspects as well. Okay. But before we do that, I just want to illustrate the issue that you're facing if you decide to become a plant breeder. So often we assume that plants are easy. They're simple things to work with. But this is just to illustrate that the genomes of most of our crop species are comparable or even more complex than the human genome. Humans are diploid. Crops can have various levels of ploidy. Soybean, what does a partially diploidized tetraploid mean? Well, it means it was a tetraploid, and then it's lost some of the components, and so part of the genome is diploid, part of the genome is tetraploid. But you can see that the human genome is actually smaller than many of these, particularly hexaploid wheat. It dwarfs the human genome. And you might imagine that we have the sequence of all of these genomes, given the state of sequencing these days. But even now, we don't have a fully annotated wheat genome because of the complexity of it and trying to annotate it. There's so much repetitive sequencing there. So this is what you're working with if you're a plant breeder. Complex genomes and different ploidy levels. But really, if you are looking at breeding, this is the sort of pipeline you're thinking about. This is really the cyclical pipeline that plant breeders have had to deal with throughout the domestication of agriculture. So the objective, pretty simple. What you're trying to do is introduce a trait into what we call your elite line. So your elite line is normally the line that you're working with. It has a number of desirable traits in it already, and you've identified a novel trait in another variety that you would like to introduce into this. Okay? So an important point about plant breeding is, as I stated here, it's not mechanistic. So this is a really important point to take about plant breeding, and it's hammered home to me about 10 years ago when I was talking to a plant breeder. And I said, oh, that's a really interesting mutant you've got there. What's it mutated in? And I said, don't know. I said, what, you don't know? No, it doesn't matter. It's just a trait that we're interested in. We don't need to know what is the underlying mechanism here. We just need to know that this is a trait that can provide some benefit to us, so we're not bothered about what it actually does. There are issues associated with that approach, of course. But you can see, you're identifying a trait in a line of interest. You're going to combine it with your existing line. There are always issues here. Anyone imagine what there's an issue here if you're trying to breed in to an existing variety? If you want to breed two things...
Would you lose like the desirable traits?
Well, yeah, absolutely. Good. That's very good. We'll come to that in a second. It's very simple. You've actually got to be able to breed them. They do have to be compatible. The next step is to identify the progeny that actually have the trait that you're interested in. That might not be all of the traits. This is just the trait that you're interested in. And this is genetics. We've just looked at the genomes, various levels of ploidy. So this step here isn't going to happen on the immediate progeny necessarily because of the genetics. If you're thinking about a diploid organism, your immediate progeny of a cross are hybrids, they're F1s. They'll be heterozygous if it's a diploid. If your trait isn't dominant, then it's not going to express itself in this first generation. So you have to think about that. So you might be working with your second generation. And next, you've got to test your progeny for the trait of interest. And we'll talk about various ways of testing, but it might be a phenotypic test. So it might be based upon a phenotype that you can observe and score. And then generally, if you are a plant breeding company, this will be done over multiple sites because they have to inform the growers of how these particular lines will perform under different environments. Because not everyone's growing things in the same place. So this just gives you an idea of scale. So this might be a typical breeding pipeline, and you'll start with quite a large number of plants. So in this instance, 150,000, over 10 different locations. They might be locations within the same country, they might be different countries. And at each step, each subsequent step, you're sampling this population for the traits that you want, selecting those plants, and then collecting the seed, and they then move on to the next step where you'll test obviously fewer individuals but a greater number of different sites. And this goes down and down and down until you're testing at multiple sites with a small number of individuals. And this is all before you can even think about deploying to the market. So this can be up to 10, 15 years of work, depending upon the crop, depending upon how fast you can generate it. And in terms of things, this is just for maize. So I said they're scored. This is what maize breeders would be scoring. So they score them at multiple stages of development for multiple traits. So just think about that page there. 150,000 plants for all of those traits. That's a huge amount of information, but it's also a lot of work. So plant breeding is an awful lot of work. So these are pipelines that are in place by big seed companies. So this is just to give you an idea of scale and what the objective of plant breeding is. And now we're going to talk about, briefly, the Green Revolution, because this has had massive consequences for ourselves and for the planet. So I think Professor Gray introduced you to Norman Borlaug the other day, who won his Nobel Prize for his work in the Green Revolution. And So what we're talking about, to give some context, is that the Green Revolution happened between the 1950s and really the 1980s. And early 1900s, etc., you had suddenly the input of the Haber-Bosch process, which allowed you to start to utilise high-nutrient fertilisers as well. By the mid-1900s, breeding technologies were, quite advanced. So they had these pipelines, they had these capabilities. But the problem at that time is that many countries were having to import grain in order to sustain their populations. So there weren't actually many countries in the world that were net exporters of major crops. And we're talking here wheat, rice in particular. So it was around about the 1950s, 60s that Norman Borlaug was tasked by the US government to take on their wheat breeding program and to generate higher yielding varieties of wheat. And he actually did this in Mexico. So the reason he chose Mexico was because of its environment. He recognised that he could take his breeding program and perform it in two different sites in Mexico, which was in the north and also, I believe, in the Midlands as well. Now, what this enabled him to do was to cycle between these sites. Within one calendar year, he could actually grow 2 crops. So again, time is always an issue with breeding, because think about how long it takes for a plant to develop. This isn't microbiology where you can go and inoculate something and you've got a culture the next day that you can work with. This can be 6 weeks, 8 weeks, 3 months, 4 months, 6 months before you have a product. So utilising these 2 sites meant he could advance the breeding program really by half. He could half the time. But the significant thing in reality, which possibly wasn't realised at the time, was that actually these 2 sites had differing photoperiods and differing environments. But this was significant for wheat. Wheat at the time was what we call a long day flowering plant. So in order for it to flower, to set grain, it needed increasing photoperiods. So what we are seeing at this moment in time, if you planted wheat now, it would be growing during increasing photoperiod. The day lengths would be getting longer. And that's what traditionally wheat needed. And what Borlaug was doing was actually planting in the north when the day length was getting shorter, and then cycling to the next site, which actually did have a long day when the day length was getting longer. Now for many plants, photoperiod is very important for controlling the flowering process. By cycling between these 2 sites with their differing photoperiods, what he was able to do was actually break this nexus and generate photoperiod insensitive varieties of wheat. So they're going to flower.
What do you actually mean by cycling between the sites?
So you grow them in the north, where the photoperiod was getting shorter. So you're going to be selecting for plants that are going to be flowering under those conditions. You're then going to cycle to the second site where the day length's getting longer, and again, you're only really going to select for plants that are going to set grain in those conditions. Normally, you would need to only be growing in one site and not the other for them to set grain. So you're basically selecting for mutants that are insensitive to the change in photoperiod.
You mean like taking the seeds and then replanting them?
Yes. Yeah. So yes, in all of these instances, you're collecting seed material. So that was one of the major things, breaking that photoperiod nexus. One of the problems with the Haber-Bosch process was that fertilisers absolutely increased productivity. But for wheat, they generated an issue. They actually resulted in a process called lodging. Anyone heard of lodging? Some of you heard of lodging. So lodging is where essentially the grain head becomes so heavy that, particularly if it's windy as well, it'll cause the stem to break. So you'll actually get stem snappage. So lodging results in actually large crop and yield losses. And this was all because these plants were being grown under high nitrogen and fertiliser use. So their stem elongation was incredible, grain production was incredible, but the stems were snapping. And so Borlaug looked into this, and this was one of the major innovations. He scanned the world for varieties of wheat that could help alleviate this issue. And he identified a variety called Noren 10, which was a dwarf variety of wheat. Now again, he didn't care why Noren 10 was a dwarf variety of wheat. He was only bothered about the dwarfism. And using his rapid cycling methods, he introduced the dwarfism into high-yielding varieties of wheat. And that really was the nexus for the Green Revolution. It enabled, once these were new lines were exported throughout the globe, and the same process was applied to other crops, I might add, it resulted in certain areas of the world, Asia in particular, becoming actually net exporters for the first time. So this is just an indication of what happened over the course of the Green Revolution. And there's some significant points to point out here. So really, don't worry too much about these being two separate lines. You've got yield and production increasing year by year due to these methods and lines being introduced. But significantly, seed size wasn't increasing. So if seed size isn't increasing but yield is, it's telling you that your seeds are bigger. And importantly also, the area of land wasn't being being farmed wasn't increasing either. So you're actually getting more yield off the same amount of land. And we can see here in grapevine actually, the impact of dwarfism. So you have a normal variety here, dwarf variety here. This is actually involved, the genes involved are involved in gibberellin biosynthesis. Gibberellins are often involved in stem elongation and germination. But you can see the difference in stem morphology here. And because the plants weren't having to put so much of their resources into stem elongation, those resources could be diverted into grain filling. So that's why you're getting bigger seeds, because they didn't have to invest so much into their stem growth. Now, whilst the Green Revolution has enabled us to a certain extent, enhance our food production, there are of course consequences of that. Our current world population would not be what it was, or what it is, if it wasn't for the Green Revolution. And you can see that as in many lights. The Green Revolution obviously uses an enormous amount of fertiliser, which has impacts upon the environment and global warming. But at the same time, it has meant that actually a huge amount of land has been saved from being converted to agriculture. So it's far from a cut and dry, positive development, but certainly there are balances to this. So that's just taking you through the Green Revolution and how Borlaug was using these breeding technologies to generate these high-yielding dwarfed lines. That was quite a rapid process. I mean, he was really only tasked with this in the 50s, and this was all coming onto market by the 60s, 70s. It's not always the case, so there are limits to traditional plant breeding. So, Cornell University developed Potato Cultivar New York 121. So, it's resistant to a particular fungus. They had to do this by taking varieties, wild varieties in Peru, introducing them into varieties that were suitable for Northern Europe. And it took them 30 years to develop this line. So 30 years to get to commercialisation. That's an academic's career, really. One of the limitations of breeding is you are, as I said, limited by your ability to actually cross your varieties. Now, one of Norman Borlaug's dreams was to introduce rice rust immunity to all crops. So rice rust, as you might imagine, infects rice, but certain varieties of rice are resistant to it. But it also infects other crops, such as wheat. You can't cross rice and wheat, of course. The resistance genes don't exist in wheat. So you have a barrier there for being able to introduce a trait, a cross-species barrier. Traditional breeding also had unintentional consequences. So remember, you're scoring for traits. You're scoring for the trait that you're interested in. And it is genetics. That's all it is. But often it's phenotype-based. Traditional plant breeding was phenotype-based many times. There are troubles associated with this. Whenever you cross something, when you cross two species, your initial generation is heterozygous. So you have a trait that's introduced. Do you know what other traits are being introduced with it? Do you understand what other traits are being introduced with it? And this is an example here where people didn't. So this is an example of southern corn blight infecting maize in the 1970s in America. Now, what much of the maize breeding community had been doing at the time to develop their lines was to use cytoplasmic male sterile lines for their breeding program. And so they had this line called CMS-T, cytoplasmic male sterility line T, that they used for the breeding. And why did they do this? Well, when you're breeding maize, as with anything, you have to have the female parts of the plant and the male from your donor. What you don't really want to do in breeding is have your recipient line fertilised by itself. So many plants are self-fertilising, so you want to avoid that. And this cytoplasmic male sterility line helped because it didn't produce pollen. So it avoided a whole process that breeders had to go through, which was called detasseling. They had to go into the field and actually remove all the anthers, the developing anthers, from the maize flowers before they could do the crossing. And this was time-consuming, it was expensive, so they had these male sterility lines. They utilised those for the core of their breeding programme, which was great, saved them time, saved them money. But unfortunately what they didn't know was that this particular mutation, and it was actually a mutation in a mitochondrial gene, made those lines susceptible to a new variety of southern corn blight that started to appear in the late 1960s. 80% of the breeding pipelines utilised this particular male sterile line. So they were all susceptible to this new variety of southern corn blight. And between this late 60s and 70s, 15% of the maize yields were lost because of this, until they developed new varieties, until they understood why they were seeing this. So there are major issues involved if you don't fully understand the traits that are being transferred between your progeny, if you're just focusing on transferring your trait phenotypically. So what we're going to do now is look at how molecular workers come in and help with this. So we're going to look, hopefully, over the next 20 minutes, at a couple of processes. So we'll start with marker-assisted breeding and hopefully go on to mutation breeding and tilling, which are to a certain extent related. So, breeding, it's genetics. What the seed companies have excelled at, the plant breeders have excelled at over time, is developing genetic maps, linkage maps. So they will know where on the genome a particular locus is that controls a trait. And I was very careful there with my wording. I said locus, I didn't say gene, because actually, in some instances, they still don't know the gene that is regulating the trait. Again, they might not be interested, but they know that the locus is in a particular position on a particular chromosome. Now, if you have that knowledge, then what you can do is you can identify markers that are tightly linked to it. And when we're talking about markers, in this context, we're really talking about DNA polymorphisms, sequence-based markers. Often these can be ones that you can utilise via PCR. So if you do a PCR on different varieties, you'll get different sized products. But the variety of different markers available to you. New sequencing technologies means that you can now sequence lines and utilise that information to develop markers. So markers that are specific to your variety, you can map them on the genome so you can have precise information about where the marker is. And you can have this for multiple varieties before you ever do your crossing. And the advantage of this is that it allows you to select for progeny that only eventually have the trait of interest transferred into your elite line without having significant contributions from other parts of the donor's genome. So this is just illustrated here. So what you're doing in this instance is you're attempting to introduce this trait, which has been mapped to this position, into your elite line. Okay? So we're doing a cross between a donor and our elite line. And obviously in the first generation, it's heterozygous at the locus of interest. Heterozygous at all loci. What you do is, what you're trying to do through this process is eliminate as much of the donor DNA, donor genome as possible, whilst maintaining the trait, the loci that you want. So what you do is you do a back cross to your elite line. And then you look at the next generation, the progeny from this, and you use your markers to follow this. Okay? And what you're doing each time is you're looking for crossover events, and crossovers where you start to lose part of the donor regions are closely linked to the trait of interest. So you're following this loci. You'll have a marker for this loci associated with the donor. You'll have a marker with this loci associated with the elite line, and you're following the progeny through each generation with those markers. And again, you back cross to your elite line, you continue this process. And if you do this, by the time you've done 6 back crosses, you've eliminated 99% of your donor DNA. 1% of a genome is still quite a lot. So the more back crosses you do, the more you can eliminate the donor genome, to the point where you can just have your elite line with this small region of donor DNA now introgressed. This is the phrase we use, introgressed into it. So if you do have detrimental traits associated with other parts of this chromosome, through this process you've gradually eliminated those. So that's what molecular markers have enabled us to do. The other major factor is scale. So everything is made better by increasing the scale, in molecular biology anyway. The limitations for breeders was the ability to analyse their lines. But seed companies, they have pipelines, they have machines that actually do all this. So the number of progeny you can analyse is increased tenfold, hundredfold. And what that means is purely from a probabilistic outcome, you're more likely to get the progeny you want if you start with many more progeny in the first place. So one of the things I've said is plant breeding doesn't rely on you knowing anything about the gene. You're interested in a trait, you're interested in a loci that you've mapped. And this is just an example of this. So this is the development of sub-1 rice. So we often associate rice with lots and lots of water. But the fact is, rice is not submergence tolerant as such, certainly not the high yielding varieties. And what I mean here is that it's not tolerant to long periods of being fully submerged. But there are varieties of rice that are. So once the water recedes, they'll survive. And this is determined by the sub-1 locus. Now, for those of you interested, it's an ethylene response factor. But actually, the sub-1 line, the trait was identified about 50 years ago. And by around about the 1980s, there were some lines being introduced that had the sub-1 loci introduced into the high-yielding varieties of lines. But they weren't particularly great. And this was because the marker density wasn't very high at this point in time, so it was hard to follow this by marker-assisted breeding. As we developed our understanding of the rice genome, more markers were developed, and so actually the sub-1 line, a high-yielding variety of rice, the Swama line, was introduced by the late 90s. The sub-1A gene was only finally cloned and characterized in 2006. That was published in Science, a high-impact publication. And the point I'm trying to make is that the breeders had actually already introduced this trait into high-yielding varieties before the gene was ever cloned.
Sorry, can you do complementation with plants then?
Mm-hmm.
Genes identified by cloning?
Yeah, absolutely. Yeah, yes, complementation, it's standard genetic procedure. Yes. Does everyone know what complementation means? So if you have a mutation, if you have a mutant line that's showing a defective phenotype, the way that you confirm that a particular gene is responsible for that trait is to take the wild-type copy and reintroduce it into the mutant with the expectation that it should then complement, generate a wild-type phenotype again. So that's the standard practice for confirming that a gene is responsible for a trait. So, some of the processes involved with breeding. Now, I'm just going to try and cover this in the last 10 minutes or so. One of the things that I've mentioned a few times is that, of course, there are limitations to breeding. You're limited to your essentially traits that are in existence, traits that can actually be bred into your line. For many crops, this isn't just relating to your elite lines. This could actually also relate to wild varieties that can still be bred with these high-yielding lines as well. So you can utilise wild varieties to introduce new variants, new alleles in. But you are still limited by the presence, the existence of these alleles. So there's this concept of the pangenome for many species. That is the total genome size for a plant species. And actually, if you look at any variety, its pangenome for the species is much greater normally than actually the genome of an individual variety. So this is just what I'm saying down here. So you're limited by your existing alleles. So what do you do? if you have a limitation, well, the ideal process is you start to make new ones. And this isn't GM, and this is the critical point that I want to make. What we're going to be talking about next is not characterised as GM at all, except possibly in France, because there is currently something that's going through the courts that might change this. Mutation breeding, what do I mean by this? Well, this, really, if you've done any genetics, is something you should have been exposed to on numerous occasions. The idea is that you will take a variety and you will expose it to a mutagen and then screen it for novel traits. And this is exactly what's been done with crops. And actually some of this stemmed out of how to have a beneficial use for nuclear radiation after the Second World War. So obviously people were quite rightly terrified by what had happened. So to put a positive spin on it, they had to come up with ideas of how they could utilise nuclear radiation for other purposes, and one of these was to actually extend it to crop development. So they take seeds and expose them to nuclear radiation to generate new varieties. And there are numerous varieties out on market that have been generated via mutational breeding. Not always nuclear radiation, some of it is chemical mutagens. EMS is a common one there, so ethyl methensulfonate. But basically anything that can damage DNA has been used to generate new crops. And like I say, the process is expose the seed to the mutagen and then screen them for a trait that you're interested in. And like I say, critically, it's not classified as GM. You're not introducing any transgenic DNA into plants via mutagenesis means. And this has connotations for the whole CRISPR argument, which we'll talk about next week. There is a problem with it, of course, in that mutagenesis is essentially random. You can't apply X-rays and determine which part of the genome that's going to be damaged by those X-rays. You have no choice here. So it's untargeted. And what I'll do for the last few minutes is tell you a way that you can still utilise this in a targeted way. Okay? So what I'm going to do is tell you about tilling. So tilling is called targeted induced local lesions in genomes. And this was a method that was developed in Arabidopsis. So has anyone heard of Arabidopsis? Hopefully second years have been introduced to Arabidopsis before. It's a model dicotyledonous plant. It's basically a weed, but it's used by developmental biologists because it's rapid cycling and has a relatively small genome that's easily manipulated. Now, the problem here was illustrated in this example here. So imagine you're working with wheat and you're talking to someone who grows barley and they say, oh yes, we've got this variety of barley that's resistant to this particular bacterial pathogen. And we know that this is due to a mutation in this particular gene, this R gene. We've got the sequence, everything we know about the mutation. Now you're working with wheat and this resistance doesn't exist in the population at present. But you look at the sequence of the genome and you realise that that homolog is present in wheat. It's just there isn't a mutant of it. So what you want to do is see, well, I'd like to make a mutant of this and then test it to see if it will be resistant to this particular bacterial pathogen. But I can't use GM mechanisms. Okay, so I'm not going to use GM for this. So what do? Okay. So tilling is a reverse genetics process. So what I've said there is, the first thing, for tilling to work, you must know what gene you're targeting. You have to know the sequence. But it's a big undertaking because what you have to do is mutagenize a population, and now this becomes a probability problem. If you mutagenize something, how big a population are you going to have in order to identify mutation in the gene of interest, in this R gene? So think about that going back to the genome sizes. How many are you going to have to do this for? But you mutagenize this population, and then, and this is where the scale issue comes in, you have to analyze that gene in all of your progeny. You're going to scan it in all of the progeny to try and identify a line where you've mutated your R gene. And as I said, because it's using mutation, it's not GM. And it was only introduced into Arabidopsis in 2000, but it's been used in several crops since. Doesn't have to rely on mutation, by the way, because you might have natural variation out in wild species. So if you know that there is an allele that exists in wild species, you can use that information. So this is a process called ecotilling. But just to take you through how you do this. So how are you going to identify mutants? So you're going to use a mutagen, EMS or sodium azide. So this is just saying that EMS generally causes a GC to AT transitions. So often, if you think about the context there, you can often get stop codons associated with EMS. And sodium azide, AT to GC transitions. So you go through this process. You mutagenize your plants. You self them. So what you need is M2 here, because you're trying to generate homozygotes. And then the really big part of this is you have to extract DNA from all of these plants. What you're going to try and do is genotype them for your gene of interest. So you extract the DNA from all these individuals. This is an awful large number of DNA samples. So to make your life easier, you pool them. You obviously know which DNA, which plant is associated with a particular pool. And then you PCR your particular region from those samples and you analyze it. So you're mutagenizing, you're pooling, and then you're PCRing up your region to analyze it. Now, when this was originally brought out, the mechanism for identifying lesions was based upon heteroduplex formation. So what they did in the original application was they took their PCR DNA, so you've got lots and lots of DNA fragments, some of which may have mutations in your gene of interest, and they mixed it with wild-type DNA from the same region, melted the strands, and then allowed them to anneal again. And the probability is that if you have a lesion somewhere and you introduce the wild-type region as well, that you will get heteroduplexes formed. And heteroduplexes can be recognised by a particular nuclease called SAL1, which will cleave them. And what you'd do is then you'd analyse all of your products in this way. Very labour intensive. So you can imagine that now there are faster ways of doing this. So you don't need to rely on this nuclease, this method. Some of them still rely on heteroduplex formation. So some of them are using conformation sensitive capillary electrophoresis, for example. This looks at the melt temperature of heteroduplex DNA versus standard homoduplexes. But of course, if you have the money, if you're a seed company, a major seed company, you can actually just sequence all of these PCR products. And you'll be able to identify pools where there are mutations, and then you can focus in on those pools and start to look at individual plants so that you can identify the single plant that had a mutation in your gene of interest. And this is just to give you an indication of some of the instances where this has actually been used. Particularly, you'll notice this is associated here with many instances it's associated with starch. So amylose or amylopectin are components of starch, and depending upon whether you are wanting your product to be associated with food production or whether you're wanting it to be associated with, for example, wallpaper paste production, you want to change the amount of amylose or amylopectin in your grain. So particularly from the food perspective, you're more interested in something that's high in amylose, whereas from the glue perspective, you want something that's high in amylopectin. So that's why they've developed lines that are mutated in enzymes that regulate the amount of amylose and amylopectin. So what I've tried to do today is just take you through normal breeding processes. These are going on, Monsanto, Syngenta, etc. They're all utilising these methodologies to develop new lines. And the point I'm trying to put across is you don't need to know the genes to do this, but the more knowledge you have about the genomes, the more markers you have, the denser your genetic maps, then the better your objectives will be achieved. But it's done on a huge scale. You have to analyse huge numbers of plants to do this, and it means that actually new varieties are not, there's not huge numbers of new varieties being brought to the market every year. So most major seed companies will only be introducing a couple of new varieties every year because the pipeline is still 10 or so years to develop them. Okay, so that's just to go over the outlines again. And tomorrow we'll focus our attention onto more GM mechanisms. How do you introduce DNA into plants? Okay, thank you very much.
So Jeff has introduced CRISPR.
We'll talk about CRISPR in another lecture. There are various issues associated with CRISPR, and CRISPR can't do everything. It's not the golden ticket that everyone says it is.